My name is Marimina Hardy. I'm from Ghana, and I will be your host for today. So a brief introduction about myself. I'm a third year DPhil student in molecular and cellular medicine at the University of Oxford. And my work focuses on, immune responses in viral hepatitis B with a lot of computational and wet lab components. So I consider myself a virologist in training. And so I must say that this session is particularly special to me because today's guest, Dr. Peter Quashie, is someone whose work I truly admire. As a young scientist aspiring to make meaningful contributions in virology, I often look up to Dr. Quashie's approach. His depth of knowledge, his commitment to advancing molecular virology in Africa, and his mentorship of the next generation of researchers that has been nothing short of like inspiring. And I was even telling him before the call a few names of people who he has truly inspired, including myself. So about Dr. Quashie, he is an associate professor of molecular virology and the deputy director of research at the West African Center for Cell Biology of Infectious Pathogens, which is WACCBiP, at the University of Ghana. It's quite a mouthful, but I got there. He obtained his bachelor's in biological sciences from the University of Ghana and went on to pursue another bachelor's and master's in molecular biology at Concordia University in Canada, and later earned his PhD in experimental medicine from McGill. So, to our wonderful audience of students, early career scientists, and enthusiasts joining us today, thank you so much for being here as we delve into the remarkable journey. I'm really excited about this of one of Africa's most leading molecular virologists. So, Dr. Quashie, I don't know if you noticed it, but it's such an honor to have you with us today. And thank you so much for joining my science journey.
Thank you very much, Nina, and thank you, My Science Journey, for inviting me. I mean, I don't know what to say after that quite flattering introduction, but it's my pleasure to be here.
So I want to first of all address like the elephant in the room. Congratulations on your new role as an associate professor at WACCBiP. I actually saw the LinkedIn post a week ago, so this is all very surreal for me. So you've gone from, running experiments yourself to now shaping research direction at an institutional level, and that's quite a transition. How did you adjust your mindset moving from, being hands-on scientist? I mean, you are still a hands-on scientist, but to being more of a strategic leader?
Well, I think I'll start a little bit. I'll go back a little bit before I come to your question. Um, so for me, I've always wanted to do science, and I've always wanted to do um, virology. I got inspired quite early on, and I told somebody that during the pandemic, when it was all locked down and we were scrambling, collecting samples, trying to do testing, I said that this is what I had envisaged for my future, right? And I mean, I think it's good to try to understand who you are and what really drives you. And for me, curiosity has always interested me, and also being able to sort of have an impact and have a positive impact has interested me. And I was very fortunate. I always tell people that my path has been a bit circuitous. Even though I knew I wanted to do research, I knew I wanted to um, sort of have an impact in outbreak scenarios. When I was really young, I didn't know how to do that. I thought you had to go to med school or something. And then part of me wanted to be an engineer. Um, but I somehow managed to find myself doing enzymology, where I really focused on enzyme processes in microbial systems. And then I grew up in Botswana, which is ravaged by HIV/AIDS. At the time that I was a student there, I remember in science class when our teacher said, there are 10 people on the bench, 5 of you have it, and it's quite a jarring thing at that point. But then when we were doing, I think, secondary school um, diversity of organisms, so I think that's what the course was. And we learned a little bit about the virus. And then I thought, oh, if you can target the viral envelope, maybe you can block entry. And then I think somewhere in that same year, I went to buy, I grew up in Botswana, I said. So I went to buy what we call fat cakes, and it was wrapped in paper. And it was paper from the Journal of the um, East African Medical Journal. And it was talking about T20, which is a fusion inhibitor.
Yeah.
So at that point, then I started telling my friends that, oh, I think this is exactly the idea I had in mind. Of course, I didn't know that T20 is not going to get widespread use because of toxicity and all those things. But then that told me that I have something, because I got that from one class. And I said, okay, I'm going to cure HIV. That's what I said in secondary school. And then everybody had a laugh. And somehow I ended up in the lab of the late Mark Weinberg, who really revolutionized HIV research in Canada and around the world and in Africa. And during that process, there were a lot of gaps that I could see in Ghana and Africa. And because I always want to make an impact, I didn't think that life is about just being comfortable and having your family and just doing your bare minimum. I saw the difference during my postdoc, where you have undergrad, there's somebody who's really understands what the problem is really has started from trying to mobilize grassroots, and I was also fortunate early on in my PhD. I met Professor Tumbi Ndungu uh, from South Africa, and I asked him, why did you leave Harvard, where you did your PhD, to go to South Africa? He's Kenyan. And he said, well, this is where the disease is. This is where the problem is. So I looked at it, and I always remember that. And when I was writing my writing up, I looked and I saw that West Africa has a very unique epidemic. And in fact, West Africa has a very unique virology background. So that's one of the reasons why I wanted to come. And also why I really wanted to drive research on the continent, because the other thing is that you look at the field of HIV, there's fantastic drugs, absolutely fantastic drugs. And when I was a PhD student, somebody diagnosed at age 20 in Canada, a man diagnosed at age 20 in Canada has a life expectancy of 80. A regular man has a life expectancy of 75, right? But in Africa, you can't get those drugs, right? So part of it is that the research really has to happen on the continent if we want to see the benefits of that research. So that's one of those things. And then I was fortunate that I was looking around, and I met Prof. Awandari, who's the founding director of WACCBiP. And I met him in December 2013. I was in Ghana on holiday, and the the center had just been founded, as in like the grant had been won, nothing had started, but he was the first guy I saw with a vision to really do research, like proper research, um, in Ghana. So we started communicating, and then, as you say, the rest is history. So to get to your question, I am still, one thing that has helped is that we're young, um, and we're a nimble organization. but that also means that there's a lot of structure that needs to be built. And I arrived in Ghana almost, I always say that everything happens for a reason. There's a reason why I met who I met today and I didn't meet them yesterday or something. I came to Ghana in 2019, and the pandemic arrived in 2020. In fact, it arrived on my heels because I was in the UK in I think February. I had just come to the UK to the Crick in February for 2 weeks. And the day I was leaving the UK was when the first cases were coming in from Hong Kong.
That's interesting.
And then we had I had my first funding in Ghana was from a Crick African Network Fellowship. And we had a network meeting in March. Actually, I think we finished on the 10th of March 2020. And we went to the labs of the National Influenza Control Program, which was then doing the testing. It was my mentor who was leading that testing, Prof. William Ampofo. And we actually showed the guys from the Crick the PCR traces showing that, okay, these are this sample, blah, blah, blah. And it was, and that was, I think, yeah, that was 6th of March. And then a week later, we had our first cases. And Jonathan Stoy, every time he sees me, he shows me that picture he took of the PCR trace when we actually had 0 cases. So I arrived at just in time to really focus on building the teams for the response that we had. So I always say that I've benefited from wartime type promotion. And along the line, I've been lucky that I've been in I think 4 research institutions over my career. And this is an opportunity for me to try and build infrastructure, build institutional strategy. It's a different mindset, because as much as I'm a PI, and that's still a huge part of what I do, now I also have to think about it from the institutional viewpoint. So when colleagues are complaining about overheads or something like that, I have to more often than not, wear my other hat and say, well, we need to make money for the center, we need to keep the lights on and things like that. And yeah, sorry, that was quite rambling.
No. Because it kind of answered a lot of questions that I had before about why virology, etc. So no, this is perfect. But as you were speaking, I noticed that, the goal started from wanting to find the cure for HIV. And I know you've studied, HIV resistant pathways, prior. But then I also noticed that you sort of pivoted to SARS-CoV-2 after the pandemic. Um, and I've read a bit of your work, and of course, with Anna working as your research assistant, I know that your studies were, focused on immune responses and finding therapeutics for SARS-CoV-2 during the time. And that is a really, really inspiring. It was a really, really inspiring time for many of us in the field. Would you say your focus has sort of evolved a bit now that you are sort of exploring other epidemic-prone RNA viruses, or is HIV still your, main
So,
focus?
so it's very interesting. You mentioned that I've worked on a lot of things,
Yes.
but you only see the things that ends up being published. I always tell people that I did a lot of mass spec during my master's, but we didn't publish it, so it's not
That's
on.
true.
But I I believe I'm on a natural journey, driven um, by curiosity, driven by the questions, and driven by the environment. But it's very strange that you end up getting to where you set out to get to. As in, when I wrote my first draft of the title for my PhD project, it was basically looking at the resistance mechanisms of these integrase inhibitors But I had other questions that were quite interesting to me,
Yeah.
drug discovery, entry inhibition, and we did all of those things. But at the end of the day, I found it interesting when I was writing my thesis that it was the fundamental question that I started with that I ended up answering, which was heavily focused on integrase inhibitor resistance. And it's the same thing that we've gone into COVID. A lot of my funding has been driven by COVID, and I find myself now doing more and more immunology than I used to. In my undergrad, I did a lot of hardcore biochemistry, and I did a little bit more of that during my PhD, went into cell culture, virology, and dabbled with structural biology in my postdoc, but still around HIV. And now, what I think SARS-CoV-2 helped is to really build the strength of my immunology research. And serendipitously, I'm back, I think about 75% of the work we do is back in HIV. And back in cure research. I've never really been in cure research, but it's also linked to why I came to Ghana. We're doing a lot of HIV-1, HIV-2 co-infections, looking at how HIV-2 is affecting the immune response, doing antibody work. And actually, this is also related to my postdoc, because when I went to do my postdoc, I I chose to do my postdoc in structural biology because I used to do a lot of modeling, and we had a patient-derived virus that we had sequenced, and we had seen a series of mutations. And then when I modeled it, it was for the first time the active site was remodeled, and I did it like 1,000 times, different software. It was remodeled, but I mean, we published it, but I don't think anybody believed it. And that's really what I set out to do, why I wanted to do the postdoc, to see if really that's what ended up happening. But during the postdoc, I learned how to work with antibodies, how to purify antibodies, how to crystallize antibodies, and that is what we do a lot of now, because during the pandemic, there was an opportunity to get into production of monoclonal antibodies, which somehow I learned in my postdoc. So it's sort of like you have to be really open to picking up skills, picking up knowledge, but it's also not to just go with the flow. I fought really hard to make sure that I'm still very focused, because you can't really achieve anything without focus. So we're still very much into RNA viruses. I mean, I have some students who work on HPV, HIV co-infection. In Africa and in Ghana, you can't be as narrow focused as you would be, let's say, in the UK, where it's like you're working on this protein within this pathway because there's real public health need. So sometimes you have to divert, but a lot of times when those opportunities come, I look for people who have more expertise in this, and I'll be like, okay, this guy, if bacteriology, for instance, even though I started out doing bacterial work, that's not my strength. So I'm just going to push it to someone there. And so to answer your question, I'm back doing a lot of HIV, more than I thought I would be. But we're also being responsive to climate change. So we're doing a lot of, increasingly so, a lot of looking at zoonotic viruses with zoonotic potential. So we have a project coming on board that's through the Africa Pandemic Science Collaborative, which is in partnership with MasterCard and the Pandemic Science Institute at Oxford. And that's to look at viruses of zoonotic potential in bats. And we have a climate sensitive grant through the Africa Climate Collaborative, which is looking at trying to build an arbovirus surveillance system in Ghana. So those are parts of the work that we're doing here. But it does surprise, and it does seem like we're going back towards the path of HIV cure.
That is really, really interesting. As you were speaking, I kind of remember like the parallel. So my supervisor is actually on the Rio trial, um, trying to cure HIV with antibodies, and I'm trying to do a similar thing for hepatitis B with antibodies as well. So when you spoke about, antibodies going back to HIV and like relating it to SARS-CoV-2, it really hits home. So I'm very happy to hear that, and I think after this call, I'll probably be in touch just to learn more about, the work with HIV and how the antibodies are being structured, etc. Because that's something I'm personally very interested in. Um, so you spoke a bit about, collaborating with the Pandemic Institute, with MasterCard on your climate initiative. So I am curious, what principles guide you when you're sort of building these equitable partnerships, especially in spaces where, resources and priorities can be a bit different or may vary?
Okay. So I think um, in answering these questions, I'm trying to be very cognizant of your audience.
Yeah.
And when I have opportunities to mentor, I always tell people that the most important thing that can help you is to know who you are and what your motivations are and what drives you, what stimulates you, what depresses you. And I think a lot of the way I work is based on the fact that on my father's side, I'm the youngest of my generation. And so what that means is that when you have family gatherings, you have to wait for everybody to speak, you know. So you absorb everything that everybody's speaking. And I realized when I was very young that sometimes if there's an issue, I have an idea and I just wait to see if somebody's going to say it. And if nobody says it, that's when I speak up. So a lot of times I just listen, and then I try to, after listening and knowing what I want out of the collaboration, let's say a lot of the times these days we are reactive to requests for collaboration, or if it's a call, if it's something that we're trying to go in as a consortium. So I listen and I try to, within that discussion, find out what their motivations are. And then the most important thing is you have to talk clearly about what your strengths are, what you can do, what you bring to the collaboration, because one thing that I took for granted when I was in Canada, which I learned very fast when I moved to Ghana, is that competence is not guaranteed. Like, as in, like, in Canada, I would write a paper. I know these are the flaws of my paper. And I would plan experiments that, okay, the reviewers are going to ask for A, B, C, D, blah, blah, blah. And you submit it. And sometimes it comes back, they haven't asked for any of those things, or they've asked for one of them and you've prepared 10. So when I moved here, one thing I realized is that we, during the pandemic, the first grant we applied for was an EDCTP emergency grant, which was basically to do everything that we did, and we didn't get that grant. And the comment It was a collaborative grant. We had a guy from uh, IRD who happened to be stationed in Ghana at the time, who was one of our European partners. We had collaboration from the UK, but it was lockdown scenario. And I wrote it the way I would normally write a grant. I didn't take pictures of the facilities. I just said we have these facilities, we have these things. And one of the things that struck me was the reviewers said it's is it's not clear how they can execute this grant since the northern partners cannot be on the ground. And there were lots of questions about whether we have adequate biocontainment facilities, whether we have. So for the first time in my life, then if I go to a meeting, you know, I have to show that I'm competent. But I think I've also been lucky because when I was in Canada, I wasn't in Canada as an African student. I was there as a Canadian student. So I was really immersed in the culture. So I have a lot of self-confidence, let's put it that way. And so that is how I think when I approach these collaborations, I look like you have to be very confident in what you have to offer. And I've been in meetings where I'm like, I don't know what I bring to this, and I will tell them that I don't see, like, I see why maybe you want me to be part of this, but I don't see what I bring to this. And if I'm not bringing anything, I feel like I don't need to be there. I have evolved a little bit because I realize that you need a balance. You need to have those projects where you're leading. You need to have those projects where you're participating. And you need to have those projects where you're just a passenger. Otherwise you can't get that. You're going to burn yourself out. And I just learned that recently that yes, you do need sometimes, I'm sure you've done group work where it's like one person's doing all the work.
Yes.
You
Very
know,
common.
I've been in a community when I was doing my master's and PhD in Montreal. I was in this Quebec Black Medical Association youth initiative. And when I was the internal coordinator for the youth team, one thing I learned was I think that's around the time the movie Inception came. So I always used to say like I learned Inception because I would have an idea. I realized that to get things done, you have to do everything yourself. But to bring the team along, I try to make people feel like, okay, this part is your idea and this part, and then that's how you get everything done. But you can't do that for everything. Sometimes you have to have teams where you're just a passenger. So it depends on the approach. But the key thing that I would say is really success is about learning who you are, you know, and sometimes um it's just fine not being the smartest person in the room or not being the most accomplished person in the room. You just have to figure out a way to be comfortable with yourself as much as possible.
That was actually very powerful. And I think that's something that I needed to hear. And it's something that a lot of, upcoming scientists or people in our early career journey actually need to hear. Um, so that would take us a bit to, because now I'm trying to get a lot more advice from you because now I'm taking notes. These are things that I'm going to implement in my everyday life. Um, so I mentioned this at the start of the call, but that was before the live stream started. You have mentored so many emerging scientists across Africa, even my best friend, and indirectly me because she wouldn't stop badgering me about the work that you do. So this has been a really huge source of motivation for me. what advice would you give young researchers who want to move beyond good science and grow into, impactful, purpose-driven leadership positions? Like what advice would you give us? And I just want to remind the audience to start to put your questions in the chat as we will start getting to them soon.
Hmm. So I'm thinking of how to phrase this because I was going to start by saying follow your passion, but I also hate that term, follow your passion. Um, the key thing is, what are you doing right now? What do you enjoy about what you're doing right now? What do you want to be doing? And you might not have any answers for these questions when you ask them. But I always focus on trying to do the best I can in what I'm doing right now. Um, I I think growing up in a quite a competitive household, when I go to it, every time I'd get to a lab, the first time I mentioned that in family setting, I'm watching, observing. So I remember when I went to Mark's lab and I was looking, I was like, okay, lab meetings, you're quiet looking. I'm like, okay, this is the darling boy. This is the guy who knows all the stuff. He's got and then you try to find a niche where you can thrive, where you're comfortable, where you can make an impact. And the times when you're maybe going to be down, I think there was a period of time where I wasn't as productive, so then my mentality was, okay, let me try and do one thing a day. If I go into the lab, if I can just have one thing checked off, that's it. But planning, thinking a lot, reading, you know, and also letting your hair down, unwinding, not getting too stressed. People always get surprised when I tell them I watch TV. I mean, when I'm on the weekend, I'm like, I watch a lot of TV.
We need to take a page from your book.
No, you need to unwind, find something. When I was a grad student, I used to watch Kardashians.
Oh, okay.
Something that you don't have to take too serious.
Yeah. Well, that's a good one. That's a good one.
And
Okay.
then
Mhm.
and then you have to sort of look. So it's really depends whether you guys are in the UK. So I think sometimes also being cognizant that you're an ambassador for your community, which when I was in Canada was always a constant thing. Um, Now I don't do a lot of uh, I had to remind myself that I used to do a lot of community work now because of it's the community work we do is more part of the institution. But try to make an impact. It's a lot about asking questions of yourself. Doing There's a lot of resources out there. I mean, all the aptitude tests, all the behavioral tests, all those things. I've done them all, you know. And the whole point, and sometimes you do it and you don't like the answer, you do it a different way. It's just about learning about yourself. And once you know yourself and you're honest with yourself and you're honest with situations, I feel like everything sort of falls together. I'm not, and you also have to always remember that everyone is human. So when I go to a meeting, for instance, and I feel intimidated, I know that I'm not the only one who feels that way,
Yeah.
you know, and that sort of helps you to to um to become comfortable and to sort of drive change. And and then when I become too comfortable in a situation, then I feel, once you become comfortable, you become complacent, you become a little bit lazy, then you need to change things up. You need to move. You need to go somewhere else.
Yeah.
You know. Right now, I mean, it's not a work in the park, being in Ghana, doing what we do, but it's a real challenge that we have, and that motivates me. I have one goal that I'm trying to, aside from my research goals for the institution, I have one thing that I think we really need to do. And I told somebody that once I achieve that, then I feel like then I can go and do other things that I want to do, still within the same
Yeah.
sphere, uh, and mission and vision, so to speak.
Okay. That's amazing.
I don't know if I answered your
No,
question.
you did. You did. Um, so looking ahead, if we fast forward 20 years, what does success in African scientific leadership and health innovation look like to you?
Okay. So now I'll wear my other hat.
Yes, I wanted to take the research hat off.
Yeah. So I think right now I look at WACCBiP, and we've been doing interviews the whole week for graduate students, and I made the statement that this is the best graduate training program in Ghana and probably in West Africa, and one of the best in Africa. And I really believe that. It has a lot of problems, but the problems are based on the developmental stage that we are, um, as a research science ecosystem. And I think we're just building the capacity, the human capacity. We're building the critical mass of scientists. So right now, the success, that if you evaluate the success we've had, is about the number of trainees we've had, the success of our trainees, you know, the skills, the real skills that we've managed to build, and um, through people who are inspired to start up their own companies. You look at Imachi, you know, and WACCBiP is 11 years old. If I look 20 years into the future, what I think this looks like is we need to be really driving innovation. We need to be really, there should be tens of companies, startups, biotechs, pharma. We should be making our own drugs. There's opportunity. In Ghana, we have a huge herbal industry. And most of those are half-assed. Oops. I don't know if, um, uh, apologies to your audience. They're like halfway done, right? Now, when you build that human capacity to take things to the next level, then they can feed into those industries and transform them from pseudoscience
Yeah,
uh, initiatives to real science things. That's where you start to have drugs coming through. You start to have innovations, um, you know, and you start to have more South-South collaborations, more South-South impacts. The And as a center, like, I think by that time, I would expect that this would be like a really strong powerhouse. I was looking at, I think last year or something, I was like, okay, now it's probably time to start focusing a lot on IP,
yeah,
you know, thinking about projects where you can have real innovation as opposed to Me Tooism. And yeah, and
yeah,
then hopefully local funding, um, local, probably pharmaceutical driven funding.
yeah. I think it's very interesting that you mentioned innovation because I have also noticed that there's been a huge shift in the scientific community towards innovation. And I think that that speaks to, like you said, what the next 25 years would look like for the continent. And it looks like when you took off the researcher hat, you might have sold a lot of people on about WACCBiP because we have a question on how somebody can get an internship at WACCBiP. People are curious, how can they join? Maybe you could give us information about the graduate programs and how people can register for that.
Okay, so we have a number of graduate programs. I'll start with, okay, WACCBiP is sort of like we have this training pyramid structure. We do a lot of internships. These internships could be people who have finished first degree and they're doing national service. So, in Ghana, national service is a thing. But at WACCBiP also, we have what we call graduate interns. So they apply apart from the normal national service, you apply and then it's a competitive process, mostly by GPA and your research experience. And then you would be assigned to faculty to do 1 year research internship. Then we have people who maybe send an email to either myself, the director, or to the administration. They want to do internship for maybe 2 weeks, 3 weeks, 1 month, and then that would be based on availability to absorb. We
Yeah.
do some more structured ones for some of our sister universities. Every year they bring a number of students and then they will intern and then you basically grade their success, their progress. Then we have structured programs. Okay, tied after the internships, we have short workshops, bioinformatics, imaging, sometimes that is facilitated by our staff. Sometimes we have collaborators who come in and do those specialized training so you can get exposure while you're here without necessarily having to go beyond. Then we have our postgraduate programs, which are, we just started the first in-country MSc bioinformatics um, program, and this is the first in Ghana and one of the few in Africa. We have MSc Biochemistry. Because WACCBiP works in symbiosis with the Department of Biochemistry, Cell and Molecular Biology, that department actually runs our programs. So biochemistry has MSc Biochemistry, MPhil Molecular Biology, and then PhD Biochemistry, PhD Molecular Biology. And then our core WACCBiP courses are MPhil Molecular Cell Biology of Infectious Diseases, and then PhD Molecular Cell Biology of Infectious Diseases. And then I mentioned the MSc Bioinformatics. So all of those are run through the department. But if it's internship, you can send an email or a letter to WACCBiP admin, and it usually finds its way to my desk, and then we take it from there.
It's interesting because as you were speaking, I remember that I actually benefited from one of your bioinformatics workshops with Thomas Otu.
Oh,
From the University of Glasgow, yes. Actually, that actually informed me choosing the University of Glasgow. So WACCBiP has played a very significant. I'm now realizing how much WACCBiP has played a significant role in my life choices, actually. So to another question, oh, they are curious about the HIV cure. Could you speak a bit about the current HIV cure you mentioned earlier? So I'm guessing they're talking about art. You mentioned it being antibody based. But I'm not.
okay. Um, so basically that's where we're going. When I joined, there was another guy who joined Noguchi from University of Washington. Um, University of Washington, I believe Missouri or Louisiana, Professor George Che, and he had gotten funding to set up the um, HIV cure infrastructure project, which is shortened to HCRIS. And we collaborated particularly on recruitment, and then, in HIV field, the questions that still remain are few. So we saw a lot of synergies, and we started working together. And I mentioned that my background, um, I had done drug discovery, drug mechanisms, but some of the unpublished work that we had done was looking at host restriction, host pathogen interactions. And so somehow, because of this antibody work, I got this very talented PhD student from Kenya who just submitted his thesis, Edwin. And he was really interested in looking at the interaction, HIV-1, HIV-2 interaction. And that was one of the reasons I had come back. But then when I came to Ghana, I realized that HIV-2 is very scarce. So it's really hard to do that work. And then this is also part of what I say, chance, or I believe fortune, not chance, really plays a role. I was contacted because there's a Chinese company that has a new diagnostic kit, and they wanted to be able to differentiate HIV-1 from HIV-2, rapid diagnostic kit. And so at that time, we didn't really have a budget for recruitment. And so I gave them a budget, and as part of it, we used that money to aggressively try to recruit HIV-2 pure infections, and we've gotten a decent amount given its rarity, about 20 something pure HIV-2 infected individuals, and then 100 co-infected individuals, and then a few hundred HIV-1 people. We have their metadata, and then we've done, first off, started by doing the characterization of the cohort. And what we've done is done some in vitro work to show where there's a little bit of inhibition of HIV-2 on HIV-1 progression, because HIV-2 doesn't progress very fast, and we think that in some cases it self-resolves, right? Those who study viruses know that as a virus, HIV in general is a wimpy virus, but that's also its strength, right? It's because it's a wimpy virus. It sticks around until the immune system is exhausted, and then when it picks up. And but what we've seen is that HIV-2, for instance, there's very little active transmission because it's very much lower pathogenicity. So we've been trying to understand that, and what we've done so far is we did the in vitro work, and then we looked at the immune parameters between the co-infected individuals, the HIV-2 pure, and a lot of this work hadn't been done in a lot of detail because HIV-2 is less than 1% of it's around less than 5% of the global pandemic, right? And then there's not much active transmission, so that you don't have large cohorts. I think the largest cases are in Guinea-Bissau, which it's very hard to do helicopter science in. So generally I use that term deliberately for historical research reasons. So not much work has been done on HIV-2, and then the thought was that co-infections were rare, and so not much work has been done on that. And now we have the samples to do that work, and so the work we've done so far has not been done in much detail before. And where we are going with this is the impact on T cell response is not as strong as you'd expect. So I really believe it's an antibody driven response. And so what we're doing is now with the technology that we built, the capacity we built to do monoclonal antibody production, is we're going to try to produce monoclonal antibodies from the B cells of these co-infected individuals and then the HIV-2 pure infected individuals, and then go to the thing. So this is basically the next step of where we're going. And in the field of monoclonal antibodies, as far as I'm aware, there's no HIV-2 directed monoclonal.
Mm.
And then the other thing from a curiosity standpoint is that in a co-infected individual, are you going to get antibodies that have dual specificity? Where would those antibodies bind? And I think these would have a much higher potential than what is on the market now. Because the other side of this coin is that HIV-1 that is in Ghana is mostly circulating recombinant form CRF02AG, which is one of the hardest viruses to neutralize.
Yeah.
It's also one of the hardest to grow, which we're struggling with in the lab. So whatever we get basically is going to revolutionize the field, because that's also one of those fields that hasn't been done much because it's not part of a big
Yeah.
pandemic. And in Ghana, most of the research has been malaria. Virology is not a big thing here. There's a few virologists, and then it's really mostly has been about doing the low-hanging fruits. And what we're trying to do is not just do the low-hanging fruits, but we want to do the whole tree. Because really, I believe that that's the only way that you can have an impact on these epidemics here. And
Yeah,
yeah,
I actually, as you were speaking, I have like two more questions, but we're running out of time, so I might just squeeze the two in anyway. So you were speaking about, monoclonal antibodies, and I am sort of curious as to why you haven't, I'm sure maybe you have, but why have you considered broadly neutralizing antibodies instead? Because, with how
yeah,
HIV is, the virus is very prone to mutations, etc. With monoclonal antibodies, is the goal to find multiple that targets multiple sites? Wouldn't it, or what if we use BNAbs, which is like broadly instead? And that's just like a question that just came off my head as you spoke.
yeah. So I used to have this frustration with one of my grad students every time I'd say highly potent, and then she'd write broadly neutralizing. And I, you don't, broadly neutralizing is more of a public health response,
I see.
which makes sense. It makes sense that you develop antibodies that would have the broadest specificity as possible. And what we've seen in the field of broadly neutralizing antibodies is that it's almost impossible to get that. And the ones that you get that are broadly neutralizing, they don't neutralize AG, they don't neutralize HIV-2. So my focus right now is to get highly potent uh, antibodies for those hard to neutralize, and then look at the breadth of neutralization secondary.
I
It's
see.
not the primary.
Yes.
I mean, the idea is that you wouldn't use one anyway, you'd use a cocktail.
Yeah. Yeah.
So I feel like in terms of, even if you think about traditional HIV cure strategies, we need to rethink the way that clinical trials are designed, because one of the first trials in pre-exposure prophylaxis, the first, that is it, R144 trial, it actually failed. And then they went back and they looked at the data and they sort of dissected the data and they saw that, oh, it worked in this subset of patients in these, and then that's what reinvigorated the field of pre-exposure prophylaxis using treatment as a block for transmission. So sometimes when we look at it like, okay, you've given the population this drug, did you meet your set points? And if not, you throw it away. No, you need to go in and see why is it that, because there's a certain percentage of people that it worked for,
Yeah.
right? So when I think about cure, I think we are going to need a number of cures. Not one, it's not possible. It's just like cancer. It's not possible to have one cure because every virus is different. And then in virology, we know that it's a conversation between the virus and the human, you know.
Yeah, I think that also explains, why there's such a huge push to like personalized medicine as well. But yes, so I have a final question for you. So if future generations were to describe your legacy in just one sentence, what would you hope they say?
I used to have a tag line for myself in grad school, which was
Yeah.
more of motivation, and I had it on the back of my Man U jersey, and that was illustrious.
Okay, that's a good one. That's a really
Because
good.
it describes, uh, I had a student who asked whether, oh, do you want to get a Nobel Prize? And I thought about it, and I'm like, I don't know how to answer that because that's not the goal. The goal is to try and get
Yeah.
something that makes an impact. But I guess if you look at it, if you get a Nobel Prize, that means that you've done that. But that should not be your goal. The goal is to, I really want to, not from a narcissistic viewpoint, but I feel like if I'm able to have a legacy where it can be written about, it can be talked about, then I've really made an impact. Um,
That's true. That's true.
can I say one other thing
Yes, yes, please. Yes.
for your listeners is that the hardest thing that we struggle with as academics is humility and ego and when to, you know, confidence and not trying to be arrogant and stuff. If you're in science and you want to be in science and you want to do research, you want to be an academic, you're a nerd, right?
Yeah.
So you have to own your nerdship and then you have to be confident in that. And you have to learn to be comfortable with that confidence. And that's one of the hardest things to do. So, and then try to find that balance. Getting that confidence, getting that self-knowledge is, I think, the single most important thing uh, to do. The balance is hard if you don't get that balance, that's the difference between you not getting a grant or an interview because they think you don't know what you're talking about because you're trying to be humble, or you're not getting it because they think you're arrogant and you're ignoring shortcomings and things like that. So I've said it several times. I think the most important thing is to try and know yourself.
Yeah. Yeah. Yep. 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 Dr. Quashie, thank you so, so much for such an inspiring and honest conversation. For me, you've reminded us that, collaboration begins with listening, understanding what drives others, and being clear about the value of what we each bring to the table. And it's really, really powerful to hear that competence should never be taken for granted. And that success isn't just about, titles and outcomes, but about learning who you truly, truly are. One of the things that really resonated with me were your words about, sometimes being the passenger, not always leading, and always staying grounded in what we enjoy. To be honest, it's been a while since I watched the Kardashians, but I think after this call, I probably should get back into
I
it.
don't recommend it.
It's too late now. Um, But yes, I think the overall message you were trying to tell us was, um, just to be able to find time for ourselves. And, as young scientists, we're always chasing the next thing. It's always best to be grounded. Um, and another thing that resonated with me was your quiet philosophy of consistency, in that we don't have to have everything figured out all at once. We just need to do one thing a day. And that would take us closer to being, the kind of scientist, leader, or person that we are, or we want to become. And I often find that even me, like as I'm sitting here now, I'm thinking of like an experiment next week. So it's good for me to be grounded. This was such an important message for me. And to everyone listening, I think Dr. Quashie's message leaves us with something deeply, deeply personal to reflect on. We might not always have the answers, but if we stay curious, if we follow our passion, if we do our very best where we are, that in itself is progress. And the last note for me is to try and find balance between being humble and arrogant. So, Dr. Quashie, I just want to say thank you once again for sharing your journey with us. And to our audience, thank you for being part of my science journey. Don't forget to like the video, leave your comments, and share this with scientists, researchers, or just anyone who you know that this would resonate deeply with. So, Dr. Quashie, thank you. I can't say thank you
Thank you
enough.
very much, Miriam, and thank you, Ruth and your team for inviting me, and it's been my pleasure.